basically turned it against the PKR-EIF2 alpha interaction by stealing this and now making it a mimic to block the very interaction that it was born from. So this can be a very important defense system, and this happens all the time in the course of viral evolution, where they steal host genes essentially to block the very cell processes that those genes participated in. What's remarkable here is that this particular strategy has been adopted by 3 separate lineages of double-stranded DNA viruses that have each stolen their own version of EIF2 alpha to essentially block the same process of PKR. So just highlighting not only that PKR is such an important block against these viruses, but how important this particular strategy of stealing EIF2 alpha has been. So we were very intrigued by this example because this poses a pretty severe challenge to PKR's kinase domain, which is to maintain the interaction with EIF2 alpha, but somehow avoid the interaction with the viral mimic, which is only under constraint to look like EIF2 alpha. So we started off by looking at the evolutionary constraints that shape these 3 players, starting off with PKR. And what was really surprising to us when we looked at PKR in this sliding window analysis of replacement synonymous rates is you can see that if there was no selection acting, you'd basically see a DNDS of 1, this dotted line, but every single domain of PKR actually evolves under positive selection. This is really remarkable because this is the only kinase in the entire human genome that has any signature of positive selection. Indeed, the closest relative of PKR is the EIF2 alpha kinase called PERK, involved in the unfolded protein response. And you can see the overall signal is much below 1, which is a signal of purifying selection. So what about EIF2 alpha? You could imagine a situation where PKR and EIF2 alpha would collaborate in terms of their evolution to keep away the mimic that is basically trying to look like EIF2 alpha. It turns out that that would be a great strategy, but that's not what occurs because EIF2 alpha is so important in translation that not even a single amino acid change has been permitted in EIF2 alpha in 60 million years of primate evolution. So now, if you think about it, I'm sure there's a German version, but this is a game of rock, paper, scissors that you play as a kid, except the stakes are really high, and one of the parties is always playing rock, right? So now you have a really difficult problem because K3L would say, well, maybe I'll just play rock and take my chances. It doesn't really have an incentive to change once it's acquired this optimal mimic state. And so this would seem to be an almost insurmountable problem for a host gene to overcome. But the fact that we are all here in this room suggests that maybe there are some adaptive landscapes that PKR can use by virtue of its positive selection. The other prediction that comes from this interaction is if K3L could actually evolve to an optimum mimic state, it might now be evolving under purifying selection. If that were the case, we can look for that again using a histogram of DNDS, but now focused on vaccinia and smallpox proteins. So I just want to remind you, here's again that host primate histogram I showed you, where EIF2 alpha is on this side, PKR is on this side. It turns out if you look at K3L, it is the fastest evolving gene in DNDS ratios in poxviral genomes. So it is not the case that K3L has evolved to an optimal mimic status that it can just stay there. It is basically locked in a true arms race with PKR, and we became very fascinated in trying to understand the molecular basis by which PKR might be able to manifest itself in this arms race. So we turned to yeast, which does not seem like a really intuitive way to do this, but part of this was a practical problem, which is that pox viruses encode about 200 open reading frames, each of which is basically locked in a known or unknown conflict with some host protein. So taking the PKR system within a vaccinia virus system became genetically somewhat intractable. So what we decided to do, take advantage of work done in Alan Hinenbusch's lab with Tom Dever, who took advantage of the fact that EIF2 alpha is so slow to evolve that if you put human PKR into yeast cells, it'll bind and phosphorylate yeast EIF2 alpha and cause a growth arrest. In the same system, if we now add K3L, we can basically rescue growth. So now we've turned this interaction between PKR and K3L into a very simple yeast growth assay where we can now introduce mutations and ask what is the net effect of this three-way interaction. So that's what Nels did. He took a panel of primate PKRs and put them on galactose control. On glucose plates, this is simply a serial dilution that convinces you that we can convincingly grow yeast in the lab, because PKR is really not turned on here. But as soon as you put them on galactose plates, you can see that there is dramatic growth inhibition. That actually immediately tells you something very important, which is despite the very, very rapid evolution of PKR, all of these alleles have basically conserved the property functionally of binding and phosphorylating eIF2 alpha. Indeed, all of this growth inhibition goes away if you mutate the eIF2 alpha phosphorylation site. So now the interesting experiment becomes when you now introduce the antagonist K3L in this system, and when you do that, you basically see a very binary response. In some instances, like in the case of GIBBON, introduction of vaccinia K3L completely overwhelms GIBBON PKR and restores growth to these yeast. In other cases, like in human and orangutan PKRs, the growth inhibition goes on unimpeded despite the presence of PKR, the K3L antagonist. So this is exactly like those two states I described to you in my cartoon example, where the host is either winning, like in the case of human and orangutan, or the host is losing, like in the case of GIBBON, in terms of its basically interaction surface. So this is a very species-specific interaction. This is actually exemplified when we go back to vaccinia cells, infections in primate cells here. So you've got human, gibbon, and orangutan fibroblast cell lines, which we've infected with either wild-type vaccinia or a vaccinia version that has been deleted for K3L. And what you can easily see here is deletion of K3L basically had no fitness consequence in human and orangutan cells. And that's because that version of K3L is basically ineffective against the human and orangutan version of PKR, but is very effective against gibbon PKR, which is why when you delete K3L, you basically have more than a 10-fold drop in fitness just by virtue of deleting a single gene. So again, emphasizing that in these rapidly evolving arms races, these are highly dynamic, and if you look at a single host species, like people have previously focused only on human, you can miss actually very important interaction determinants of pathogenicity. So what is the molecular basis of this escape? We don't actually have the full story, but I'll share with you what we have found. And a very important clue came from this beautiful co-crystal structure of PKR's kinase domain with its substrate eIF2 alpha from Frank Siccheri's lab. And what Frank's lab showed was that the single most important determinant of this interaction is this alpha helix called the G-HELIX. If you take a closer look at this alpha helix, you can see that there are 3 residues that make direct contacts with the backbone of eIF2 alpha. Now, if you can put your biochemist intuition hat on and remember the fact that eIF2 alpha is completely frozen in evolution. By virtue of that, you might suspect that these 3 residues would also have to be frozen in evolution simply to maintain their binding to eIF2 alpha. But actually, that is not what we find. These 3 residues are the fastest evolving residues in the kinase domain, which means every primate PKR binds eIF2 alpha, but every primate PKR has a different combination lock to bind essentially the same protein surface. So this is this incredible and very unexpected diversity exactly at the host-host interaction interface, which is not what you'd suspect, unless these changes were also beneficial in the context of overcoming mimicry. So we tested that by going back to our human and gibbon example. Recall that human is completely resistant and gibbon is completely susceptible to K3L. So in the gibbon backbone, we simply introduced 1 or 2 changes from the human alpha G helix into gibbon. And what's nice is that we basically get 100-fold better protection of the gibbon PKR with just a single amino acid change from the human PKR against K3L. So this basically means that these arms races can have profound selective coefficients because this single amino acid change, if it occurred in the population, would confer such a huge advantage to this version of PKR that it would quickly sweep through the population, exactly like what you'd expect in a positive selection paradigm. So this also told us something rather disturbing, which is we were clearly not, we had part of the story but not the whole story, and that's because orangutan PKR is also able to defeat K3L, but the orangutan G-HELIX was not capable of defeating, of conferring this protection to the gibbon backbone, as you can see here. This led us on a 6-month slice and dice journey to basically figure out what made orangutan protected against K3L. And at the end of that journey, we again arrived at a single amino acid residue in a helix alpha E here, which is actually 2 helices away from this interaction alpha G helix. But nonetheless, this is actually dominant and epistatic, which means you can switch gibbon from phenylalanine to leucine at this position and gain complete protection. And if you do the reverse change in orangutan, you basically lose all the protection you had against K3L. So this change is very important, and the structural basis for why it's important is something we do not currently understand. The other thing we don't understand is that this change is not under positive selection. However, this is toggled between leucine and phenylalanine essentially throughout mammalian evolution, almost as if these two amino acid residues are the only residues that are actually permitted at this particular position, but changing from one to the other and perhaps back again can completely change the repertoire of your escape from these mimic antagonists that are encoded.